This, of course, brings us to my favorite group of macromolecules, the fats. Oh, do I like the fats? Anyway, this also includes the oils and waxes, which I'm less interested in eating, although olive oil, of course, delicious. Fats, lipids. Let's get back to really what we should be talking about, which are lipids. Lipids are great long-term energy stores. Ounce per ounce, fats and lipids contain far more calories than carbohydrates, the next best energy store. Um, it also is a structural component of cell membranes and all kinds of other stuff. So remember, you need fat. You're made of fat. Yeah. Okay, so lipid basics. Also, they are also composed, just like carbohydrates, of carbon, hydrogen, and oxygen. Difference is going to be that they have these great long chains of carbon and hydrogen, and just a few oxygens. So the ratio there of carbon, or excuse me, hydrogen to oxygen is going to be much different than that of water. We can talk about different kind of like family groups. These are loose classification groups like the fats and the phospholipids and the steroids. These are, like I said, different very loose classification groups that we use. It is important to note that we are not talking about polymers, even though they have those long hydrocarbon chains. We don't consider lipids carbons. They are big, big, big molecules, though. These are really, really huge and enormous. The fats that we're going to be spending most of our time talking about are fats that are made from one single glycerol molecule and then three fatty acids. Glycerol molecules are three carbon, one, two, and three carbon, what they're called triols, and that's because each carbon here has an OH group attached to it. The fatty acid itself has a carboxyl group on one end and then a big long chain of carbohydrates at the other end of it. They're named based on the number of carbons and the nature of the bonds between those carbons and stuff like that. Now, those that are made this way, the three fatty acids with a glycerol, are called triacylglycerides or triacylglycerol. It does set up an ester linkage between that hydroxyl of the glycerol and the carboxyl group um, of the fatty acid, and so we refer to that as an ester linkage because it has the central oxygen here between the two different carbon species, so it looks like an ester there. Looking at lipids more functionally, they are mostly hydrocarbon chains. When we talk about hydrocarbon chains, that's hydrogen carbon are certainly nice coordinating covalent nonpolar bonds. But it does have that carbonyl group in there. But because so much of it is hydrocarbon in nature, the overall molecule is therefore hydrophobic. It doesn't mix well with water. Oil and water don't mix. So the function there, the primary function that we think of is energy storage. It's because it is a good concentrated store of energy with all of those stable hydrogen-carbon bonds. They'll yield quite a bit of energy when they are broken, when we strip those hydrogens off. They do have ounce per ounce, 2 times or more the caloric value of complex carbohydrates. But energy is not the only thing they're good for. They're also good for cushioning organs from blows and injuries. They are also good for insulating. Think whale blubber when we think of insulating against the cold, like cold water. It also creates a large thermal mass. One class of lipids we need to be familiar with are the saturated fats. Again, lipids get their characteristics largely from the length and nature of the fatty acid chains, the hydrocarbon chains from the fatty acids. In saturated fats, the carbons are all bonded to as many hydrogens or carbons as they possibly can. So there are no double bonds there between the carbons. That gives us straight chains. It allows those three fatty acid tails to kind of fit together, nice long and straight nest together. This is what we see in most animal fats like butter, and those that are solid at room temperature because they are more dense. They are able to fit together more molecules because those legs nest together so well. Unfortunately, though they are tasty, these are ones that do contribute to atherosclerosis, the building up of plaques on the inside of arteries, high blood pressure, and other kinds of fantastic cardiovascular disease. We should also be familiar with the unsaturated fats. Since the saturated fats have the straight chains, the unsaturated fats have the bent chains. This happens because there are carbon bonds that are double bonds rather than single bonds. Um, Monounsaturated fats have one double bond. Polyunsaturated fats have several double bonds between them. These are the ones that are more common in plants and fish. They are liquid at room temperature. This is because those individual legs don't nest together so well because they're the bent chain, the bends at each one of those double bonds keeps them from nesting together as well. Another class of lipids that we should be familiar with are the phospholipids. These have a glycerol, but in two fatty acids. Instead of a third fatty acid, it has a phosphate group. That is a big negative charge group sitting up there. Um, because of this charged head on it, we call that the head region, is this hydrophobic or hydrophilic? And the answer is yes. The heads are, with the phosphate groups, are hydrophilic, very polar in nature. And the tails with all those nonpolar carbon-hydrogen bonds and carbon-carbon bonds are very hydrophobic, are very nonpolar. That means that the tops of the molecules mix well with water and the tails of the molecules do not mix well with water. And this nature is going to be real important when we start talking about the structure of living things and cell membranes. So what do these phospholipids do when I put a bunch of them in water? Well, since the heads of them like the water, they're attracted to the water. They're both nonpolar, so they get along. They don't have any problem being there. It's the tails that have the problem. The tails are nonpolar and they don't like being in the water. They're not attracted to water. They like being with other nonpolar compounds. And so where can they hide? Well, they can hide in the tails of other phospholipids. And so they'll spontaneously form these structures called micelles. Where the tails all are nested together, making a non-polar area on the inside, and the heads are all on the outside mixing with the water, which of course is polar. We can also put some water on the inside of this micelle, and we'll start to form this bilayer. So we'll get two layers of lipids with the heads on the outside where the water is, and on the inside where the water is, and then the tails kind of going onto the inside of the layer, forming a kind of non-polar region in the middle. We call this the phospholipid bilayer. It is where we think cells came from, this spontaneous nature of soap molecules to form these micelles and bilayers. And these are the makeup of cell membranes and the entire endomembrane system. The last group of lipids we should talk about are the steroids. Now, steroids are a special class of lipids, of fats, because structurally they are so different from the triacylglycerides we've been talking about with the glycerol and the fatty acid basics. And these are 4 carbon rings that are all fused together. And then attached to those are going to be different functional groups in different places, and where and what those functional groups are is going to change the shape of the steroid and contribute to its functionality. The classic example here is cholesterol, and here you can see the 4 carbon rings here in the middle. And then attached to them are, here's a methyl group, and here's another methyl group, and down here is a hydroxyl group. And if you were saying to yourself, well, this doesn't look like a lipid at all, why do they call these lipids? Well, this whole region down here being all hydrocarbon is, in this case, all hydrocarbon, but in most of them it's going to be largely hydrocarbon in nature, and therefore nonpolar and lipophilic, hydrophobic. But this ends down here with the hydroxyl group. That's very polar and likes to get into the water. And so I have this amphipathic compound that has the hydrophilic part and it has the hydrophobic part. So it's going to line up just like the phospholipids are with the in this case the hydroxyl group towards the water, and then this big clunky, the rest of the big clunky molecule here down in with the fatty acid chains. And that's kind of important when these guys do make up an important part of animal cell membranes. They are also a precursor. We do use cholesterol to make our sex hormones from. They are important enough that our bodies make them. Some of us are better at making cholesterol than others. And if you're like me and lucky enough to have a gene that, or genes that allow you to make lots of cholesterol, thanks mom, and you have to be careful, and those can high levels can contribute to cardiovascular disease. But the main function here of the cholesterol that I've been talking about is to get in there in the cell membrane and kind of get in between those fatty acid legs and keep them from nesting together too close to one another, and keep that cell membrane kind of fluid and flowing, so that, remember, there's no bonds in between each individual lipid. They're just held in kind of position the same way a bubble is held together, just by the hydrophilic, hydrophobic interactions of the amphipathic molecules and the water that they are surrounded in. Sex hormones are good examples of steroidal hormones. Those are hormones that are based on cholesterol, and a good example of the importance of the functional group and the influence in the location and type of functional groups on the overall functionality of the steroid itself. Looking at cortisol and aldosterol. Cortisol is a stress hormone that influences blood flow and through capillary dilation, and like I said, it's a stress hormone, fight or flight responses, where aldosterone, certainly important, is involved in maintaining proper electrolyte balances through kidney function, through renal function. And really the big difference between the two, when you look closely, is just shifting that hydroxyl group over, or taking it out and replace, putting a little carbonyl action on there, and you end up with poof, aldosterone. The rest of the molecule is really exactly the same. When we look at the sex hormones themselves, testosterone and estradiol, which are the male androgen and the female estrogen, respectively, really the big difference between the two, the only difference between the two, is that carbonyl group in the testosterone and the hydroxyl group in the estradiol. A good example of, the difference between the two. So there are several different functional groups that you're going to see hanging off of these cholesterol-based hormones and other compounds that are steroids.